The correct use of spreadsheets to analyse scientific data is an important skill to master. In this spreadsheet, we are showing the data from a very simple spectrophotometry experiment. We have a number of known standards which we've used to generate the data for our standard curve. So we've got our absorbance readings. We've then used the average function to calculate the average of each set of values. These data can then be used to produce our standard curve, which gives us a nice straight line fit. We've got our y equals mx plus c value shown here. We can also use the slope intercept functions to generate these numbers as well, and these are shown on the spreadsheet. Now in this experiment we've got three unknowns, A, B and C. In each case we've taken a reading or a set of readings for each of the unknowns, again calculated the mean using the average function. Now when it comes to converting this mean absorbance back to the amount of substance present, we've got two approaches that we can use. We can use the approach shown here, which is the correct way of doing things in that we're referencing back to the slope and the intercept, which is from the equation y equals mx plus c. And this is cell referencing, and this is important because with cell referencing it means that, let's say we've got some of these values wrong in our standard curve. Let's say we later realise that it's not really 50 micrograms per mil here, but is in fact 5, 100, 20 and 40. As you can see, as I have changed those values, we've also had a change in the amount shown here. And this is correct. This is what we want to happen. We want any changes to be automatically cascaded throughout the spreadsheet. What a number of students will do is they will construct this spreadsheet so that the value calculated here is not using this cell referencing, but they will actually type in what I call hard code the numbers. So for example, they will put in the slope as 0.025. They will put in the intercept, which will be in here as 0.006. Now there's two problems with doing this. Yes, it gives the correct answer, but the problem is we've now hard coded these numbers in. So any changes that are made to the standard curve or any of the other values on here, so let's say a change in the concentration or a change in one of the readings, will not be cascaded down here. Now look what happens when I move back to the original values. Keep an eye on this 0.53 micrograms down here, and keep an eye on the 24.29. As you can see, as I change the values back to their original values, we get the original line coming back, we get the original intercept data coming back, and we now have back the original numbers that we had here, but that 0.53 hasn't changed because in fact the slope value hasn't been automatically updated for this cell, and neither has the intercept. This demonstrates why it's very important when you're setting up spreadsheets that you don't hard code data from one cell into a calculation in another cell. You should always use cell referencing in the form of E15 or B6, etc. That way any changes that you make that change the numbers in those cells will be reflected throughout the rest of the spreadsheet. That is, those changes will cascade.